Hello, everyone. Welcome to this set of lectures on action potentials. We're going to start off with this topic of graded potentials versus action potentials. And we're just going to go ahead and answer this question in this portion of the lecture. What are the differences between graded potentials and action potentials? So let's get started. Okay, so I just want to remind you that whenever we have ion movement across the cell membrane, that's going to cause the membrane potential to change, and that is an electrical signal, right? So we have an example of that right here. I want to remind you a downward blip in membrane potential is called a hyperpolarization. An upward blip in membrane potential is called depolarization. We have two main types of electrical signals that we get in neurons, graded potentials and action potentials. And I'm now going to explain to you some of the differences between graded potentials and action potentials, okay? So let's start off with graded potentials. First of all, I just want to draw your attention to this figure here. What we're doing is we are recording with an electrode at this location on this neuron's cell body, and it has received a stimulus from another neuron. We'll talk more about the nature of that stimulus in the next set of lectures, so don't worry about that for now, but suffice it to say that here we are getting a change in membrane potential. And this type of change in membrane potential is called a graded potential. Also, don't worry yet about these guys down here. I'll talk about them later, okay? Okay, so let's talk about some of the characteristics of graded potentials. First of all, they usually occur in the dendrites or in the cell body. Just like in this example, we have this graded potential happening in the cell body. They can be depolarizing or hyperpolarizing, right? So if we look at this graded potential right here, we can see that it is depolarizing. Can you imagine what a hyperpolarizing graded potential would look like? Go ahead and either draw or describe that on your worksheet. Hit pause if you need to. Okay. Next up, the size of the signal is proportional to the size of the stimulus. So here we have a certain size stimulus going into this neuron's cell body, and it is creating this size of graded potential. Here's a question for you. How could you get a smaller signal or a smaller graded potential? Go ahead and think about that. Write that down on your worksheet. Hit pause if you need to. Okay, next up. What is going on with my, okay. Next up, no minimum stimulus is required to trigger a graded potential. So you can have a teeny tiny stimulus coming in, and that will give you a teeny tiny graded potential. You can have a big stimulus come in, and that will give you a big graded potential. This is not the case for action potentials, as you will see. With action potentials, if you give a tiny stimulus, you won't get anything at all, okay? Graded potentials can sum. So you can take one of these, you can take another one, you can put them together, and you can make a larger graded potential by summing them together, okay? And then finally, this is kind of a point that I'm going to spend a bit of time talking about. The size of the signal degrades over distance. Okay, so now I'm going to draw your attention to all of these panels right here. What we're looking at here is we're looking at the same graded potential as it's being recorded from 3 different locations on this neuron. When we start here, we can see that the graded potential is this big, okay? That graded potential is going to travel along the cell body. As it makes it to the next electrode here that we're recording from, that graded potential has gotten a little bit smaller, okay? Not only that, but it's also a little bit later because it takes a little bit of time for that potential to reach this new location, okay? And then finally, once we get to this electrode right here, that graded potential has become even smaller and later, okay? So it's kind of like dropping a pebble into a pond. So when you drop a pebble, or throw, maybe would be better. If you throw a pebble into a pond, you get these big ripples right next to where that pebble is. As you go farther away, those ripples, they're going to travel out from that center where you put that pebble, right? But they're going to get smaller as you go away, okay, from that point of origin, okay? So the same thing is happening here. And one thing that I want to point out here is that here in this diagram, you can see that there are these arrows that are kind of getting weaker and weaker as you go farther and farther away from where the stimulus was originally applied, right? So this potential is getting smaller and smaller. But I also want to point out that it doesn't just go in this one direction. That's something that often confuses students about this figure. It actually spreads in all directions from this point here. So again, thinking about that pebble dropping into the pond, it's going to radiate in all directions from where it originally starts off, okay? All right. Here is another diagram illustrating the same concept. What you're looking at here is on the y-axis, you have the strength of the graded potential, so the amplitude of the graded potential, okay? And then on the x-axis, you have the distance from where the stimulus was applied, okay? And what you can see is that it's highest here in the middle, where that stimulus was first applied. And then as you go farther and farther away from that point, that graded potential gets weaker and weaker, okay? In the bottom here is kind of the same thing. This is actually a corresponding picture. It corresponds to this graph right here. So we have the stimulus here. We have sodium ions. I want to remind you that these potentials that we're looking at, these graded potentials that we're looking at, are a result of ion movement across the membrane, which is causing those changes in membrane potential, which are the signal that we're looking at, okay? So we have this change in graded potential, we have a change in membrane potential here. It's strong, very close to where the stimulus has been applied, and then it gets weaker as we go away, as indicated by these smaller arrows as we move away, okay? Another thing I want to say about this figure is that these numbers 1, 2, 3, 4, 5 correspond to these numbers here, 1, 2, 3, 4, 5. So that's just kind of illustrating that same concept, okay? Here is a question for you. We have these two locations, A and B. Which location has a stronger graded potential? So go ahead and hit pause and answer that question. Okay, so I'm just going to go ahead and answer that for you. Hopefully you figured out B has a stronger graded potential than A because B is closer to where that original potential was generated, okay?